Good afternoon, everyone. It is a great honor to present here today. I'd like to sincerely thank to organizing committee and Dr. Tiku for invitation. I'm Kang from Seoul National University. Today I'll be presenting on towards digital breeding in chili pepper. I hope my presentation provides useful insight into the future of pepper breeding in India. The main content of my talk will cover the three key areas. First, the concept of digital breeding, and second, the molecular breeding status of pepper, and finally, genomic selection and the perspectives of digital breeding in chili pepper. Looking at the Korean market for your interest, the cultivar types and market value show the dry pepper accounts for the largest share at 45%. Among fresh peppers, um, uh, chili pepper, hot type, takes up about 35%, and sweet pepper accounts for 17%. Let's briefly review the history of a crop breeding method following the Mendel's discovery of the law of genetics in 1866. Crossbreeding began in 1900 and 19th century, at 1900, and molecular breeding, marker-assisted breeding was introduced in 2000, and genomic selection method began to introduce in around 2010. Crossbreeding usually takes up about 8 to 10 years to develop. I'm sorry, what happened? To develop cultivar. Molecular breeding 6 to 8 years. But for digital breeding, when we combine phenotype data, genotype data, we expect that the cultivar development time will reduce about 3 to 5 years. So pepper breeding has evolved around a hybrid cultivar, and Korea was first country to use CGMS system. And during last 3 decades, many resistant cultivars, disease-resistant cultivars has been developed. And these days, um, molecular market, thanks to the molecular markers, lifespan of a new cultivar was shortened from 5 to 6 years to 3 to 4 years. And this molecular marker development was aligned with the sequencing technology development. So, what is digital breeding? Digital breeding is a technology that utilize AI-based on phenotype, genotype, and environmental big data to develop new cultivars. The core components of uh, digital breeding are, as shown here, genomic data, phenotype data, environmental data, and these data can be used uh, for predicting genotypes of breeding materials. So when we combine these data, and we can develop new cultivars more efficiently and more uh, much faster. So the decreasing cost of the sequencing has led to exponential increase in genomic data. The graph clearly shows a sharp rise in total number of published genome, including the chromosome level genomes. And this data surge is deriving the transition from molecular breeding focused on qualitative traits to digital breeding focused on quantitative traits. So, since the first reference genome for pepper was published in 2014 by Korean group and also published by Chinese group, molecular breeding research has developed rapidly through whole genome resequencing of cultivated and wild species. But single reference genome cannot represent the diversity within a species. So, therefore, the concept of a pangenome is become critical. The pangenome refers the entire sets of genes found in all individuals or strains within a specific species or clade. Therefore, a pangenome uh, integrating genomic information from diverse pepper lines is essential. So our lab is currently working on constructing a graph based pangenome as you shown here using 26 annual genomes, and we are currently finished resequencing about 300 accessions of capsicum annuum genome. So why do we need to develop more genome information? High quality genomic data is essential for accuracy of digital breeding, while GBS and array data are low density and limited for internal LB based imputation. Reference based imputation using pangenome offers rich variants and higher resolution. So high quality imputated genomic data improves rare variant detection and enhances genomic prediction accuracy. And we are planning to build haploid reference genome panel for pepper. So also the advancement of phenotyping tools is crucial for accelerating the breeding process. Phenotype can be categorized into cellular and biochemical and microscopic phenotype. Moreover, integration of image sensors, as shown here, RGB sensor, hyperspectral sensor, or thermal infrared sensors, revolutionized the way to research or collect data on plant growth, development, and response to stresses. These sensors can capture the detailed information about plant morphology, growth rate, leaf color, and other characteristics in real time and non-destructively. When combined, these genomic data and phenotype data with AI technology, this advanced analytical and phenotyping method offers powerful tools for plant breeders. So these are some of the examples we installed these days at SNU experimental farm. So we have XYG system utilizes camera, RGB and hyperspectra and thermal cameras mounted on moving rig to capture non-destructive and repeated images above the plant. Also, cabinet system monitors phenotypes of individual plants using cameras installed with a seated cabinet. And also we have a sensor system which can monitor the movement of water and nutrient to measure the drought tolerance and other nutrient movement. So this is one of the example measurement of a Phytophthora resistance using a thermal camera. So we used susceptible and resistant accessions, famous resistant accession CM334, And to make a long story short, as you can see here, these are susceptible responses, and the blue heat map is the resistant accessions response. As you can see, we could detect differences in the heat development by the resistant and susceptible lines. So we are making use of this information for more detailed phenotyping for disease resistance. So my laboratory has developed most of molecular markers used by pepper breeders in Korea. Pepper molecular breeding focused initially on the disease resistance, such as Phytophthora blight, CMV-TSWB, potivirus, and so on. And these days, we are more focusing on secondary metabolites like capsaicinoid, carotenoids, anthocyanin, and other secondary metabolites. The main tools utilized include the genetic material and sequencing technology like GBS, Lensig, and whole genome resequencing. And using this information, well, we are sorry, uh, identifying genes, controlling fruit size, yield, and secondary metabolite, and also we are using this information for QTL mapping. And these days we are using genomic selection method, which I'm going to talk later on for the quantitative trait improvement. So these are some of the examples of our work. So you can identify the published paper in Google Scholar. If you type my name, you can find all the information we published previously. Um, Traditional molecular breeding primarily focused on single gene qualitative traits. However, agriculturally important traits like yield, content of secondary metabolite, and disease resistance are under control of a quantitative trait. There are several difficulties in improving quantitative traits. So comparison QTLs across different studies is challenging. Markers are often far from QTL due to the low density genetic maps, and candidate genes underlying the QTLs are often unknown. So, but new tools like genomic sequencing technology and phenotyping technology provides new opportunities. So, quantitative trade involves so for identifying quantitative traits, we are using this platform. So we have a core collection, which I'm going to introduce. For core collection, we genotype with different sequencing method and phenotype about 30 different traits, including plant architecture, pungency, fruit color, and disease resistance. And then finally, association analysis and validations are performed with various statistical models like GRM and LMM. These are some of the examples of our genetic material. We have several recombinant inbred line developed for capsaicinoid. We measure capsaicinoid at different environment and different years. And we also have a core collection consisting of 351 accessions, predominantly capsicum annuum. But we also included several other capsicum species. And we selected this core collection from almost 500 accessions of peppers, and we quickly genotyped these accessions and also used passport data to select core collection accessions. So we are using this core collection for genetic study. And as I mentioned, these plants are phenotypes in different years and different environment, and not only for capsaicinoid, but also other traits are phenotyped. So using these phenotypes, we are doing genomic selection. Genomic selection involves building a prediction model using training population uh, of using genotype and phenotype. And by training prediction model, uh, you can apply this prediction model to a breeding population without phenotyping. Only with the genotype, we can predict phenotype. So this prediction, we call it genomic estimated breeding value. These values are used to select best variety for breeding. While this is comparison between genomic selection and mass, while mass is effective for qualitative trait or a few major QTL-associated quantitative traits, genomic selection is effective for quantitative traits and uses whole genome or genome-wide molecular markers and gives a better prediction for phenotype. So this table shows the 10 different models used in genomic selection. Genomic selection models are broadly divided into linear and non-linear model. Linear model considering only additive effect, but non-linear model including corner model and machine learning model, you can detect epistatic effect or effect of markers or marker environmental interaction. So by testing different models, we are currently making use of three models in red, as you can see here, for prediction of a phenotype. So first, we applied the genomic selection for fruit morphology. As you can see here, fruit traits include fruit length, width, weight, and pericarp thickness. So after training the prediction model, we applied the prediction model to validate the model in the recombinant inbred population. For this, we used about 2,100 SMP markers. So this slide shows
Hello, check.
the, the results of uh, uh, genomic selection for fruit morphology. These are different models. These are different traits. As you can see, depending on the prediction model, there is differences in the prediction accuracy, as you
Hello,
can see
Dr.
here. For
Dr.
some
Wang.
phenotype, you can see prediction model gives you better prediction accuracy. Also, when we tested different number of markers for different traits, even we reduce number of markers, the prediction accuracy
Dr.
did
Wang,
not
your time
affect
is
it that
over.
much.
Hello,
So
your
by
time.
testing different models and different marker sets, we were able to obtain accuracy of a fruit-related traits appears to be between
Hello,
0.70 and 0.86.
hello, Dr. Wang, your time is up.
And we wanted to test this for capsaicinoid content prediction. As you may know that
Check.
the
Hello.
capsaicinoids of a different population of
Hello, Dr. Kang.
capsicum
Wang, your
is
time is over.
largely
Dr.
controlled
Kang, sir.
by many environmental factors and many genes.
Hello, your time is
So
up.
we also tested this using same training population and same marker set, and tested population is a commercial elite line.
Hello, Kang, your
So when we
time is up.
applied genomic selection of capsaicinoid content, same as we tested the fruit-related trait, different models gives different prediction accuracy, but some prediction models commonly detected the commonly predicted the phenotype more accurately. So these are some of the results as you can see here. G-blob-RR and RKHS random forest model gives you 0.7% prediction accuracy.
Dr.
So
Kang,
to improve the prediction accuracy, we use different numbers of markers, and also we use the genome markers from genome-wide association study. So when we use the genome-wide marker, about 4,000 SNPs, RKES model gives you about 0.6
Dr. Wang, your time is
to
up.
prediction accuracy. But when we only use the markers from GWAS, much better accuracy. And when we combined these two marker sets, genome-wide marker and the markers from GWAS, most significant markers were used as a fixed effect, we were able to predict
Hello.
capsaicinoid content much
Hello.
better
Hel-
prediction accuracy, like
Hello,
almost
Dr. Kang, your time
0.8.
is up.
Finally, I'd like to mention new editing technology in pepper. So we published this work in Plant Journal this year. So this is called the virus-mediated gene editing. So pepper, it is very difficult to make a transformation. So this is one of the hurdles for genome editing, but it's possible to make genome edit the transformation. So we collaborated with a company called Toolgen, and we transformed the Cas9 gene into one of the pepper lines. And instead of doing a pepper transformation every time, we make use of this Cas9 overexpression plant, and we inoculated TRB, as shown by Professor Koeda. We are using TRB vector not for silencing but for the delivery of guide RNA. So we can do two ways. You
Dr.
can
Kang, can you
use.
please conclude as we are running short of time?
Okay, so almost finished. So we are making use of Cas9 overexpression plant and TRB vector. Now we are able to develop different edited materials in pepper. So these are undergoing the editing lines we are developing. So photo bleaching is typical symptom when we silence Python desaturase, clustering gene, seedlessness, and recessive genes like pelota we are also doing, and also we are developing capsaicinoid content. So towards digital breeding, I showed you some of the examples. We need to have more data, and the phenotype should be standardized, and genotype and pangenome should be developed better. And we need to have, to provide the information, we need to have open source platform. And most importantly, we need to validate the genomic selection or AI-based prediction model for cultivar development. I'd like to express my sincere gratitude to my lab members, collaborators, and company partners, including seed companies here, and I'd like to thank funding source. Thank you very much for your attention.